think about. And then the idea is basically to get rid of these hyperimmune-associated diseases that are a small fraction that are caused by mutation. This is something I like to show to parasitologists. Parasitologists always work on the things that kill you. There are things out there that don't kill you. We want to, if we reintroduce this in the population, we want to avoid the helminths that are not well adapted. And we want to do it only in a way where we can control the colonization rate. And of course, there's, if the patient has problems with coagulopathy, if they have a problem with malnutrition or hydration, or there's a couple of other indicators that would mean you're called contraindications. You don't need the therapy. So this is Stacy Bilbo here. I think she's a member of the RCN. She just couldn't be here because she's in Florida. She's very interested in this project. We're working on it together. We think that if the immune system gets out of whack, it can affect your brain. Not just your kidneys, not just your liver. And from a biologist's perspective, the brain is just another organ, so it makes sense. This is a comment here that strikes close to the heart of autism. We're working on a model right now to see if indeed in rats we can sort of prevent them from getting an induced autism-like syndrome. And if we can do that, I think then we'll have a huge bridge into the clinical world as far as getting these organisms reintroduced back in the population. The rate of autism right now in South Korea, the numbers just came out, 1 in 38 males is on the spectrum. And the numbers coming out in the United States that are not out yet, but I do know they're almost identical to that. So that's a huge portion of your population. If every woman has 2 children, then your chances of having an autistic child are very high. All right, that's the end of the human disease business. I'm about out of time, but you guys just had a conference looking at wild immunology, so I'll probably go ahead and skip this. Our main stuff is to, has been comparing these wild rodents to try to find out what's really normal. We published a couple of papers on it. If you're interested, I can show you that. There's a lot of work that we haven't published that we're into right now. I'll just show you one thing that looks, it's very, very interesting. If you look at this CD62L, or so-called L-selectin, this is something that helps your immune cells stick. So it helps your immune cells do their job, T cells in particular. But what happens is, if you look at wild rodents, and I know it's a small study, but the p-values are good, even with the Bonferroni correction. But what happens is, in wild rats, they're all pretty active. The cells are pretty active, and it looks like they've shed off a lot of their L-selectin, and you find it floating around in the serum. So here it is over in the serum. It's not on the cells near as much anymore. There's a lot of variation in the wild rodents. I could say a lot about that. Very interesting looking at the variation. I think the take-home lesson there is, but we're not sure yet, the immune system needs to be doing something. Maybe a Th1 kind of response, maybe a Th2 kind of response. All the traditional paradigms that we see in the lab rodents, you get a parasite, you upregulate T regulatory cells. That just doesn't happen. That's not what we're seeing in the wild rodents. In fact, a study just came out looking at mice, same deal. You don't really see a difference in the T reg cells or T regulatory cells. But that's all immunology speak. The point is, we just don't know what's going on, except that it looks like the immune system needs to be doing something to avoid the kind of pathological reactions that we see. We see changes all over the place. This is sort of an overview of the immune system. I'll go ahead and stop it right here. So I think, yeah, we're out of time. There are a lot of changes in those wild rodents that are tremendously different than a lab rat. And there's some caveats to the research. I've got a whole list of those somewhere. But we're not so much concerned about genetics as we are stress. And stress is a huge factor. We're changing our model to try to account for that.
So I can imagine when if you ever get to the point where you want to develop it into a drug, that you get this one genetically pure element that the FDA will want you to have something really pure. But would it be better? Is it going to be important to have a genetically diverse population of helminths as well? And can you see any of that in your work, or are you adding just a pure line to your?
Well, I think what we're going to have to do is do the experiment in humans to answer that question. for what we saw in the multiple sclerosis study, those people were colonized, there were 12 patients total, they were colonized with 5 different worms. And you know, the tapeworm worked just fine, the woodworm worked just fine, the other 3 worms worked just fine. So it might not matter so much. The problem is with our post-industrial society, we're making this so clean that it might not be a big deal to get back to normal. We might not have to worry that much, which is what I'm hoping for, but to answer your question, we'll have to do it in humans, I think. I expect to be bombarded, with questions. Parasitology.
There's plenty of time. Maybe
right. Okay. Maybe later, all right.
Okay, thanks, William.
Very